Hello and hi, my name is Nabiha Umairah binti Abdul Ghaffar. Today I'm going to present my bachelor dissertation project with the title of Comparative Study of Fingerprint based on Gender Identification. Here are all the contents that I will present. So, I'll begin with background study of my project. Each human fingerprint is distinct and does not change with either age or growth. Fingerprints provide an unbreakable method of personal identification. A fingerprint is a unique pattern of ridges and valleys on the surface of the fingertip. It formed during fetal development and remains unchanged throughout a person's life. A person's gender can be accurately ascertained by using certain fingerprint characteristics such as fingerprint ridges density and the amino acid profile of sweat residue found in the fingerprints. Fingerprint based on gender identification have been chosen to investigate the comparison on the crime scene. However, fingerprints are commonly used in forensic science and law enforcement for identification purposes. Alright, so now we will move to the problem of this study. Fingerprints issued to identify suspect. But there is a research showing that apart from the identified suspect, the ridge density could be used to differentiate the gender. Based on the overseas study, the gender identification using fingerprint normally used. The other parameters also like the thickness to valley ratio, depth, and type fingerprints pattern. In addition, all these factors can be affected by the type of surfaces and for how long fingerprints can be used. The significance of this study is the ability of the forensic scientists to investigate whether the fingerprints belong to male or female. In this project, the quality of fingerprint can be proven over a specific period by observing the ridge details and comparing it to the grading score system. Next, the objective of this study is to determine the ridge density of fingerprint, male and female, over time, and to evaluate the ridge density on non-porous over time based on different grading system of fingerprints. Now, let's look at latent fingerprints in this study. Latent fingerprints are usually invisible because they are composed primarily of sweat, oils, and other natural secretion from the skin. When a person touches a surface like porous, non-porous, or semi-porous, the natural oils and sweat from their skin are transferred onto that surface, forming a latent fingerprints. Latent fingerprints often contain extracellular DNA which originate from shed skin cell and other biological materials left behind when a person touches a surface. This extracellular DNA can be crucial in forensic investigation as it can potentially identify the person who left the print. Fingerprints are composed of a complex mixture of substances secreted by skin, primarily sweat and sebum, along with shed skin cells and environmental contaminants. Acrine sweat glands are distributed throughout the skin, including on the friction ridges, surfaces of the fingers, palms, and soles. These glands produce a watery sweat that contains various chemicals. There are sebaceous glands everywhere on the body. Fingerprints can also pick up trace elements and contaminants from the environment such as dust, dirt, or chemical present on the surfaces. These environmental factors can sometimes affect the visibility or preservation of latent fingerprints and may need to be considered during forensic analysis. We go to the next section where the ridge count to determine the gender in this study. The finger ridge count method involves counting the number of ridges present in a specific region of a finger, typically within a defined area or a segment of the finger. This method has been explored in various studies as a potential indicator for gender detection, though its reliability and accuracy can vary. This area is chosen because it often exhibits stable ridge pattern and is relatively easy to measure consistently. This method uses the parameter of ridge density over time and makes sure that same area fingerprints is used. We move along to the next part where the method I use. First, I use fingerprint powder and brushes. This part, I use a clear laminate film to deposit the fingerprints. Then, the fingerprints were taken from donor, which is one male and one female. The fingerprints were taken from two types, which were natural rubbing, where the donor directly stamped on the laminate film. Next was rubbing on the cheek before depositing on the laminate paper. I repeat both types of natural and rubbing on cheek fingerprint for 4 times. The fingerprints were developed by using powder dusting method. Next, all the fingerprints were photographed every 0, 24, 48, and 72 hours and analyzed using grading score of fingerprints. We move along to the next aims, which is the result. First, from this picture, show the differences between natural and rubbing on cheek fingerprints. Natural fingerprints refer to the fingerprints that are deposited on the surface through natural contact with the skin without any intentional or specific manipulation. Rubbing on cheek fingerprints was clearer as stated in this picture because they consist of sweat, oils, all the chemicals in the faces, and the other residues from the skin that adhere to the surfaces. Next, we move to the graph of quality fingerprint, male and female, for both natural and rubbing on cheek over time. Both graphs show that male fingerprint have a better quality compared to female. One of the factors that I would say, the female's fingerprint is distinguished by a high quantity of white lines that disrupted the quality. Males generally have a larger sebaceous gland and produce more sebum compared to the females. This can influence the amount of oily residue left in the fingerprints and may affect the visibility or preservation of latent prints. In the graph of natural fingerprint, the average number of qualities of fingerprint is between 2.75 to 3.25. That is lesser compared to the graph of rubbing on cheek fingerprint that show the average number of qualities of fingerprint is between 3.0 to 3.5. I would take other causes that may degrade the quality of fingerprint, which are humidity, where the moisture causes fingerprints to blur or smudge, reducing their clarity and visibility. Environmental contaminants such as dust, dirt, oils, and the chemicals can accumulate on the surfaces and obscure fingerprints residues. We move to the next part. These pictures show the square of 5 times 5 mm squared that I used to synchronize the place to count the ridges. The ridge density for male in this histogram was 9 ridges over 25 mm squared, while female was 13 ridges over 25 mm squared respectively. Moreover, the ridge count was maintained for both male and female because in this study, I used one male and one female sample. It shows that female has high ridge density because the smaller surface area may lead to a higher concentration of ridges per unit area. In conclusion, from the method of ridge density, it was successful to show that female have high ridge density compared to male, so we can determine the fingerprints of male and female over time. Last but not least, I recommended for future studies to increase the number of samples, which is the donor for both and females. Also, use a greater amount of data set over a longer period of time because researchers can have a better access the consistency and significance of any observed difference in the fingerprint characteristics.